Hi, my name is Abby Hickstein, and today I'll be telling you about my summer research in Dr. Edinger's lab at Marquette University. In the lab, we work with smooth muscle tissues. Smooth muscle is a type of muscle tissue in which the contractile fibers are not highly ordered. Cardiac and skeletal muscle are both striate in appearance, whereas smooth muscle is not. It is also associated with involuntary movement not under conscious control, and it has one nucleus per cell. Smooth muscle can be found in the gut, blood vessels, the intestine, the uterus, veins, arteries, and many more internal organs. Some of the functions of smooth muscle include moving food through the digestive tract, regulating airflow in the lungs, and blood flow in the arteries. The aorta is the largest artery in the body, which begins at the top left ventricle of the heart. The heart pumps blood from the left ventricle in the aorta through the aortic ventricle. The aorta is in the shape of a tube and is about a foot long and over an inch in diameter. The main focus of the lab's research and the overarching question that the lab is trying to answer is how can we identify the mechanisms of morbidity in individuals who have a coarctation in the aorta? Before explaining more about the lab's research, I want to define exactly what a coarctation is. A coarctation is a narrowing of the aorta. So as you can see right here is where the defect is, and then here's a normal aorta versus an aorta with a coarctation. As you can see, the aorta wall is very, very narrow. This is a congenital heart defect, meaning it's typically present at birth when a baby's aorta does not form correctly during growth as it develops during pregnancy. The coarctation blocks normal blood flow through the body, which leads to high pressure at the head and arms, and low blood pressure at the legs and the body. A coarctation can be surgically fixed to look like a normal aorta once again, but there are still health problems that can occur down the line, such as brain aneurysms, high blood pressure, heart attacks, and hypertension. There are a lot of different hypotheses as to what is the cause of coarctation and why people still have side effects after having the coarctation surgically fixed, but the answer is still unknown. Treatments for coarctation have improved life expectancy, but the affected individuals still suffer from a lower than average lifespan. This is due to cardiovascular disease and hypertension. These mechanisms of morbidity are being studied in our lab through an animal model that replicates aortic changes in humans and mimics the coarctation of the aorta as well. The summer project will be focused on using the proteins non-muscle myosin, smooth muscle myosin, smooth muscle actin, NPR3, and filamin to see how they interact with the coarctation secondary effects. This will be done by performing H&E staining and immunohistochemical staining. The immunohistochemical stain uses the antibodies for the proteins shown earlier to see if they interact with the tissue sample. H&E staining is used for microscopic evaluation to show contrast between all the fixed materials on each slide. At the end of my project, I hope to find the correlation between the proteins chosen and the coarctation secondary effects in the smooth muscle. Thank you for watching.